A muscle's contractile units lie within individual muscle cells, which are also called muscle fibers. Each muscle fiber contains numerous bundles of protein filaments. These bundles, called myofibrils, are organized into repeating contractile units called sarcomeres. A sarcomere contains two types of protein filaments, actin filaments and myosin filaments. Each actin filament is attached to a Z-line, which is found at either end of a sarcomere. The thick myosin filaments lie between the Z-lines but are not attached to them directly. Instead, they are held in position by a protein called titin. A proposed mechanism for the relaxation and contraction of a sarcomere is called the sliding filament theory. During a contraction, sarcomeres shorten. However, the actin and myosin filaments remain the same size. They simply slide past each other, changing their relative positions as the muscle contracts and relaxes. Contraction is triggered when an action potential in a somatic motor neuron reaches the neuromuscular junction, the junction between the neuron and the skeletal muscle. Depolarization of the axon terminal causes the release of the neurotransmitter acetylcholine into the synaptic cleft between the neuron and muscle. The neurotransmitters trigger the muscle fiber to fire an action potential of its own. This electrical signal propagates along the muscle fiber's plasma membrane. It also propagates into the plasma membrane's tubular invaginations called T-tubules. The T-tubules lie adjacent to the sarcoplasmic reticulum, an organelle that consists of a membranous network of compartments. The SR stores calcium ions, sequestering them from the rest of the cytoplasm. When the action potential passes through the T-tubules, the sarcoplasmic reticulum releases calcium into the cytosol. The calcium ions diffuse throughout the cytoplasm of the sarcomeres. Within the sarcomeres, the actin filaments look like two strings of pearls twisted around each other. The myosin filaments are thicker and have knob-like heads at their ends. Actin filaments are decorated with two types of proteins. One protein, troponin, lies at regular intervals along the actin filaments. Another protein called tropomyosin runs the length of each actin strand. The myosin heads at this point are bound to molecules of ADP and inorganic phosphate. After the sarcoplasmic reticulum has released calcium, the calcium ions bind to molecules of troponin. Upon binding, troponin shifts position and pulls the tropomyosin molecule aside. When tropomyosin moves, it exposes a myosin binding site on each of the actin subunits of the actin filament. The neighboring myosin heads can now bind to the actin filament. When a myosin head binds to actin, it forms what is referred to as a cross bridge. After myosin binds to the actin filaments, inorganic phosphate is released. The release triggers the myosin heads to pivot, forcing the actin and myosin filaments to slide past each other, and ATP is released. This movement represents the power stroke of muscle contraction. Before the myosin heads can release and perform additional power strokes, they must first bind to ATP. When ATP binds to the myosin heads, the heads release the actin filament. ATP is quickly hydrolyzed into ADP and inorganic phosphate. The energy released by hydrolysis cocks the myosin heads, preparing them for another cycle. Soon after the action potential ceases, the sarcoplasmic reticulum pumps the calcium that it had released back into its interior. After calcium leaves the troponin molecules, the tropomyosin molecules again hide the myosin binding sites. Actin and myosin filaments then slide back to their original positions, relaxing and lengthening the sarcomere. The sarcomeres of a muscle fiber contract and relax again with each signal from a motor neuron.